Okay, so now we're going to talk about bacterial infections, okay? And just like viral infections, first thing you're going to do is, actually just like any infection, the first thing you're going to do is you're going to mount your innate response. And the innate response is going to take over for the first 2 weeks until you have your adaptive response ready. So the first thing that's going to happen is you're going to introduce these bacteria into your body. So again, let's assume that it comes into the skin, okay? So now you have a bunch of bacteria in the dermis of your skin, and this is a bacteria right here, okay? And there are two things that are going to happen simultaneously. Let's go over the easier thing first, okay? So the two things that happen simultaneously is you're going to have resident macrophage activity, okay? And then the second thing that's also going to happen at the same time is your alternate complement pathway. Okay? So resident macrophage activity. So like I said, you're going to have bacteria, let's just say that they come in through the skin, okay? So in your skin, you're going to have resident macrophages, and those happen to be called Langerhans cells. You don't really need to know that right now, but essentially you have these macrophages, okay? And these macrophages are always here. They're just present. They're like the patrols of this region. They recognize these bacteria, and what they're going to do is they're going to phagocyte them. They're going to eat them up because that's what their job is. They're going to macrophage. They're going to, well, phagocyte them, okay? And now, again, let's represent the pathogens in a different color, so in pink. And then what it's going to do is it's going to, again, chop these up into little pieces. It's going to break up your, it's going to break up the bacteria, and it's going to present it onto both MHC1 and MHC2. Okay, so you have MHC1 and MHC2, and they now have antigens on them. Okay, and this is particularly done through the exogenous pathway because you are bringing in external material, whereas viral infections will put on antigens via the endogenous pathway because viruses infect from the inside. Okay, so this is what happens in this is what macrophages do. So they've put on the antigens on the MHC1 and MHC2, and what they're going to do is it's going to go into, let me move this all over, it's going to go to, it's going to go to lymph tissue, and the function is to activate T and B cells over there in the lymph tissue, okay? But before it leaves, before this macrophage leaves through your lymphatic vessels, it's going to release a few cytokines. And the cytokines that it's going to release are IL-1, IL-6, IL-8, and TNF-alpha, okay? These are important to remember because these are going to be important for step 3 and step 4, which we'll get to after this, okay? So I'll make these a different color. Now, as this is happening, something else is happening on a more microscopic level. So I'm going to zoom in and I'm going to show you, let's say this is the bacterial wall. This is the wall of the bacteria. Bacterial wall, okay? What's happening is in your tissue, you're going to have these proteins called C3 that are just floating around. They're not really doing much, but they will spontaneously and randomly split up into two smaller proteins, C3b and C3a, okay? Remember that C3a, actually, you know what? I'll tell you what the function of these are later. I'll just draw the skeleton of what exactly is happening right now. So C3b, what it's going to do is it's going to recognize this bacterial wall and it's going to go stick to it. So now you have C3b that is sticking to the bacterial wall. When it does this, it gains an enzymatic activity. And this enzymatic activity that it gains, is you're going to find another protein called B. B is going to come over here and it's going to be cleaved into BB. Okay? So let me draw this actually as 2 steps. So step 1 is that you have just C3b is over here. Step 1. Step 2, now what you're going to have is C3b and then BB. Okay? From big B. And then you're going to split this also into BA. So notice how the B will bind to the bacterial wall, B will bind, and A will go away. That's a good way to remember which ones are actually going to stick to the bacterial wall. Okay, so now the next thing that's going to happen is actually the step number 2 is called C3 convertase. This molecule, this dimer of C3BB is called C3 convertase. It's called C3 convertase because then what it's going to do is it's going to take even more C3. Remember, C3 was the first protein that you had? It's going to take even more C3 and it's going to split it up even more. So now you get even more C3b and you get even more C3a. And then what you do is you make a BB sandwich. So you have, remember, originally you have the C3b on the first one, and then you have the BB, and the new C3b that you just cleaved will just go and make a sandwich. So the patty is going to be the BB, and then the buns are going to be the C3b. This is called C5 convertase. Okay? And C5 convertase is going to do, I'm just going to abbreviate this, C5 convertase is going to do the same exact thing that C3 convertase did, except it's going to do it to a different protein called C5, okay? So now what's going to happen is you're going to recruit C5, you're going to split it into C5b, and then also C5a, okay? And I need to make some more space, so I'm just going to scoot this all over, zoom it in a little bit, okay? And make the wall a little longer, okay? So now once you have C5b, it's very easy from here. The rest of it is very, very easy. It automatically starts recruiting a bunch of different proteins. C5, C6, C7, C8. They just come right away. And then what you're going to have is you're going to have a bunch of proteins that make up the C9. So you're going to have 9 C9 molecules. Okay? So you're going to have 9 of these. So just pretend like this is a 9. Okay? You're going to have 9 of these, and what they're going to do is they're going to make a pore that's going to penetrate the membrane. This pore is going to be continuous with the membrane. And this is called the MAC complex. And what the MAC complex does is it literally creates a pore in the bacterial wall and in the plasma membrane, which is going to cause cellular lysis. So this bacteria. So now let's draw it on a slightly more zoomed out scale. So this is the bacteria. What happens is you put on the MAC complex. So you put on the MAC complex, and what's going to happen is then the cell is going to lyse. Okay? And when it lyses, it's going to release some cellular contents. And these cellular contents are called F-met. Okay? So now let's just fill in some details. Actually, I'm going to change the color of this to orange. Okay? Let's fill in some details. Let's talk about the function of some of these things. So aside from this, the pathway that we just talked about with C3b to C3bb to C3b, bb, C3b, to then the MAC complex, then you have some other things. You have C3a. So let me just recolor this. Oops. You have C3a. C3a acts as an anaphylotoxin. Anaphylotoxin. Okay? That's the same over here. C3a. This is the same C3a. Okay? C5a is also an anaphylotoxin. And we'll talk about what these things mean. We'll talk about what specifically that actually is. And then, okay, so you have C3b that's on the wall, but then you're also going to have other C3bs that are just floating around. So let's just say that you create even more C3b, some C3b that's not even being used. The function of this C3b, I should have drawn that in orange. The function of the C3b that does not get involved in the alternate pathway, this is going to be an opsinin. Okay, and again, we'll talk about what all of these things are. Okay, so now you have everything in orange, all the things that I just drew in orange, they're going to be reused for the next steps. Okay, so in the first 2 steps, your resident macrophage is going to release IL-1, IL-6, IL-8, TNF-alpha. In the alternate complement pathway, you're going to release C3a, C5a, and C3b, and also F-met. Oh, and F-met's function is chemotaxis. Okay, now before we move on to step 3 and step 4, which are also going to happen simultaneously, there's some important regulation steps that we have to know. Okay, from going back to your alternate pathway, you have a few steps. from step 1 to step 2, from C3b to C3 convertase. This step is going to be promoted by factor D. Okay, so green means it's going to be promoted, okay? And C3b is going to be inhibited by factor I. Okay? The next step from C3 convertase to C5 convertase is going to be promoted via properdin. And this step, C3 convertase, is going to be inhibited by factor H and a molecule called DAF. Okay? So kind of think of it like the step from C3b to C3b, the step from C3b to C5 C3 convertase is going to be promoted by D. It's going to be inhibited by A. Okay? The step from C3 convertase to C5 convertase, let me pick a different. This step is going to be promoted by properdin, and it's going to be inhibited by H and DAF. Okay? Those are important because that's probably what they're going to ask. That's probably what they're actually going to ask, is how do you promote and how do you inhibit this pathway, okay? And by the way, this is important to mention, the alternate complement pathway is also known as the properdin pathway. The properdin or alternate complement pathway, okay? All right, so now I mentioned that all the stuff that I drew in orange, these are the things that are gonna result in the next two steps, which are also gonna happen simultaneously. So what are the next two steps? Step number 3 is gonna be neutrophil, also known as PMN, recruitment. You're going to recruit neutrophils, okay? And step number 4, I'll give this some space. Step number 4 is going to be your lectin complement pathway. Okay. So the neutrophil or PMN recruitment. So what's going to happen is you're going to have a blood vessel. Okay? And along your blood vessel, you're going to have part of your blood are going to be these neutrophils, PMN. Okay? Now the first thing that's going to happen is the lining of your red blood vessels are going to increase, their expression of two proteins, okay? You're going to have selectin over here, selectin over here, and you're going to have integrin from the neutrophil side, okay? This is promoted, okay? And this is going to cause rolling adhesion, right? And this is essentially where the neutrophils are going to marginate. That means that they're going to stay onto the margin of the membrane. Okay, so right now they're floating through. They're floating through the cell, the red blood vessel, and now they're on the margin. They're on the vessel wall. Okay, the second step is going to be called firm adhesion. Now, in order for firm adhesion to take place, the vessel is going to have to express a protein called ICAM1, and the neutrophil is going to bind to ICAM1 with its own receptor called LFA1. Okay, so why does your vessel start producing ICAM1? This is where your other, these orange molecules that we just talked about, this is where they come in. Okay, so this happens because of the action of TNF alpha, IL-1, IL-6, and IL-8. Okay, so remember all of the things that the macrophage released, they're going to result in what's called firm adhesion. Okay, they're going to result in what's called firm adhesion over here. All right, and let's make the vessel a little bit. Let's extend the vessel wall just so we have more room to work with. Okay. So now that after firm adhesion, what's going to happen is you're going to get little holes in your vessel wall, and this is caused. And then what's going to happen is your neutrophils are going to squeeze through here. Okay, this process is called diapedesis. So, step 3, diabetes, and this is caused by your anaphylotoxins, C3A, C5A, and also histamines. And I didn't mention before, but whenever you get a bacterial infection, your mast cells are going to activate. Okay, so part of steps 1 and 2, in addition to resident macrophage activity and then the alternate complement pathway, is your mast cells are going to activate because your mast cells they're resident to this tissue. They're going to activate and they're going to release histamines. So these histamines, all 3 of these things, are anaphylotoxins. Okay, and all that means is that's a fancy word to say that means that your blood vessels are going to vasodilate. Vasodilation. Vasodilators. Okay, and that's how you pretty much get your neutrophils out into your tissue. So now you have neutrophils in your tissue. Okay. And your neutrophils, by the way, I forgot to mention, what is the point of bringing neutrophils into your tissue? Because neutrophils are what's going to be the primary killers of bacteria in your innate immune response. Okay, these are the foot soldiers. Your macrophages, all they do is they're just the initial. Your resident macrophages are just the initial killers. They're the first responders. But your neutrophils are what's going to do majority of the actual killing. So you need to recruit your neutrophils. You need to bring your neutrophils in. So it's kind of like the macrophages come in first, but then the neutrophils come in second, and they do majority of the work. Okay? So now you have your neutrophils that are now in your tissue, and now they have to find the bacteria. So let's draw the bacteria again in pink. And remember that we talked about over here, we talked about C3b is an opsonin, right? What an opsonin does is it opsonizes. And to opsonize something means that you're literally going to coat it. You're just going to coat it. Okay? So in addition to initiating the alternate pathway, C3b is also going to function as an opsonin. And an opsonin is going to draw the neutrophil towards it via the action of chemokines. Okay? So this step 4 is called chemotaxis. Okay? So then you're going to use your chemokines. So what are your chemokines? Your main chemokines are IL-8s, and I think that's it. Also remember we released FMET. FMET is also when you use the MAC complex to destroy the cell. FMET is also a chemokine. Okay, so your chemokines are IL-8 and FMET. Okay, so the next step then is once you've brought the neutrophil to the bacteria, obviously the function of the neutrophil is going to macrophage. It's going to phagocyte it. Okay, so now you have this neutrophil, and this neutrophil has consumed this bacteria. So now you have this neutrophil that's inside of this bacteria. Sorry, this bacteria that's inside this neutrophil. So step 5 is phagocytosis. All right. Step 6 is actually killing. Wait, 7. Step 6 is actually killing the bacteria. So it kills it pretty much via oxidative stress. Oxidative stress. Okay? And to generate oxidative stress, what you are doing is essentially you're activating the pentose phosphate pathway. And what you're doing is you're oxidizing NADPH. So you use 3 enzymes. You use G6, oh, it's weird, G. You use G6P dehydrogenase. After that, you use NADPH oxidase. And then if you want to go above and beyond, what you use is another enzyme called myeloperoxidase. Okay? And myeloperoxidase creates bleach. And bleach will literally poison the bacteria. Okay? But G6PD-H and NADPH oxidase, you're putting on oxidative stress on the bacteria, and that's going to cause it to die. Myeloperoxidase is going above and beyond to bleach the bacteria, and that's just an extra way to kill it. So if you have, there's an important clinical correlate related to NADPH oxidase, okay? If you have a decrease in NADPH oxidase, or if you have a mutation of NADPH oxidase for whatever reason, it's going to cause a disease called chronic granulomatous disease, okay? Remember that neutrophils are the main responders in your innate immune system to bacterial infections. So if you do not have a functional NADPH oxidase, then what's going to happen is chronic granulomatous disease is going to present, and you're going to have essentially your bacterial infections, like a common cold or a flu, those, or not the flu, the common cold, any kind of just basic bacterial infection that you get can be potentially life-threatening because you do not have a strong enough innate immune response to kill them. Okay? Now, if you have a dysfunctional myeloperoxidase, that's not very clinically relevant because myeloperoxidase, you can still kill the cell without myeloperoxidase. Myeloperoxidase is just going above and beyond. Okay? So this is how neutrophils are going to kill bacteria. Okay? Now, we said that at the same time that neutrophils are going to kill the bacteria, you're going to have the lectin complement pathway. The reason the lectin complement pathway happens after the alternate complement pathway is because in order to start the lectin complement pathway, you need a molecule called mannose binding lectin. Mannose binding lectin. And in order to generate mannose binding lectin, this needs to be generated from the liver. Okay? In order to generate from the liver, you need a cytokine called IL-6. So remember that we released IL-6 from the resident macrophage activity. As you can see, IL-6 is right here. So IL-6 has two functions. One function is that it's going to allow for the firm adhesion of neutrophils onto the membrane wall. And the second function is it's going to go to the liver, and the liver is going to produce mannose binding lectin. Mannose binding lectin. Okay? And just like when we did this alternate pathway, we're going to zoom in to the bacterial wall. So this is the bacterial wall. And we're going to zoom in and see what happens in the lectin binding path, the lectin pathway, okay? So you're going to have, first thing is you're going to have these two moderator proteins. They're not particularly important, but they're going to bind mannose binding lectin. And then mannose binding lectin is going to convert two molecules, okay? First thing it's going to convert is C4, and then after that it's going to convert C2, okay? And then that's essentially step 1 and 2, but I'm just going to draw it as step 1. Step 1. Okay. What that's going to do is it's going to make C4b, which means that what's going to be released is C4a. And you remember A goes away and B binds, right? That was the case for all of the other proteins, C4, C2, C3, C4, C3, and C5. But for C2, it's the opposite. C2b goes away and C2a binds. So you have C2a over here. This is the only exception to B binds and A goes away. Okay. Now after this, it starts looking very familiar to the previous one. This molecule is called C3 convertase, which is going to do the same exact thing that C3 convertase did in the alternate pathway. So what it's going to do is it's going to take C3, it's going to bind C3b, and it's going to release C3a. So what you get is now another sandwich. Another sandwich right here. Okay? And this is called C5 convertase, which does the exact same thing that it did before. It's going to take C5 and it's going to split that into C5b, C5b, and into C5a. And remember, the A goes away and the B binds, right? So you have C4b, C2a, C3b, and now the new thing that you just added is C5b. And now the rest of it is exactly the same. It's completely identical. As soon as you bring in C5b, the rest is going to happen right away. C6, C7, C8, and then your MAC complex, which is formed from C9. Okay, this is your MAC complex. And then it's going to do the same thing. It's going to cause the cell to lyse. Okay, results in lysis. And obviously when you do lysis, you're going to release even more FMET, which is going to mean that you have even more chemotaxis, which means your neutrophils can become even stronger. Okay? So just as a review of your innate response, the first two things that are going to happen is your resident macrophages are going to act at the same time as your alternate pathway. Your resident macrophages are going to release IL-1, IL-6, IL-8, and TNF-alpha. And then your macrophages are also going to present MHC1 and MHC2, which are going to go to lymph tissue to activate T cells and B cells. Okay? At the same time, your alternate pathway is happening, and that's going to result in the release of a bunch of anaphytoxins, C3A and C5A, as well as an opsinin, C3B, as well as FMET, which is chemotactic. And all of these things in orange are going to play a role in the next two steps, which is neutrophil recruitment and the lectin complement pathway, which we just talked about. Okay? So now we mentioned right here that your resident macrophage activity, it has an important function in that it's going to go to your lymph tissue and it's going to activate T cells and B cells. So while steps 1, 2, 3, and 4 are all happening, while the alternate pathway is continuing, while neutrophil recruitment is happening, while the lectin complement pathway is happening, your macrophages are busy. They're going to your lymph tissue and they're activating your adaptive response. So now your adaptive response comes into play. Okay? And just a little bit of histology just so we know where we are. We're going to be in a lymph node, okay? Now in your lymph node you have these trabeculae that are extensions of the connective tissue, and you have what's called the paracortex. The paracortex is this region right here. So this is your cortex right here, and this is your cortex right here, and your paracortex is the deeper portion of the cortex. So it'd be right around there. Right along right here is your paracortex, okay? So in this portion of your lymph, you have macrophages, the same exact macrophages that were the initial responders. These macrophages come, and we have on them MHC class 1 and MHC class 2 that are presenting our antigen of interest. Okay? That's the first thing that's going to happen. Okay? They're going to come over here. And now what they're going to do is they are going to interact with, actually, so not only are they going to present these antigens on MHC1 and MHC2, but they're also going to present antigens on their own membrane, okay? So we have, let's draw the MHC1 and MHC2 facing down. Okay, so you have these, and these have the antigens on them. But you're also going to have antigen just on the membrane wall itself. So what's going to find this is going to be antibodies, and these antibodies are part of a naive B cell. A naive B cell is a B cell that is active, but it has not ever been exposed to antigen before. So essentially the macrophage, what it's done is it has interacted with the bacteria out in the periphery, out in your tissue. Like let's say this bacteria has infected your skin. So what's going to happen is the bacteria has infected your skin, the macrophage is going to take this bacteria, it's going to phagocyte it, it's going to present, it's going to kill the bacteria, it's going to chew it up, it's going to kill it, and it's going to present its antigens on the macrophage, okay? Then the macrophage is going to take the sample of antigen, go to your lymph tissue, and it's going to start activating B cells and T cells by showing them this antigen, okay? So you take these naive B cells which are fresh from the bone marrow. Remember, these come from bone marrow, okay? Bone marrow. These come from bone marrow. Okay, so these naive B cells have just come from the bone marrow. They've never been exposed to antigen before, and these macrophages are exposing them to antigen. Okay, this antibody is specifically IgM. Okay, that's important to remember that a naive B cell, its first, the first isotype of antibody is IgM. Okay, so now what's going to happen is your naive B cell is going to activate. All right, your naive B cell is going to activate. Activated B cell. Now this active B cell, okay, this active B cell is very limited in what it can do. This is because this is a T cell independent activation. As in, this B cell has been activated independent of a B cell. So because of this, there is no isotype switching. What that means is that this activated B cell is stuck in IgM. It cannot become IgD or A or, well, no, it already has IgD. It cannot become IgA, IgE, or IgG. It cannot become any other type of isotype. The other problem is that you cannot make plasma cells or memory cells from these. So these are very short-lived cells, and they can't really do quite as much. Okay, so that's why you have T cell dependent activation. Okay? So in T cell dependent activation, what you're doing is you have a T helper cell. And this T helper cell is also naive. Okay? This T helper cell has a T cell receptor for MHC2. And let's erase the MHC1 because it's not relevant over here. Okay? This T cell receptor has specificity for this MHC2. Okay? What's going to mediate this bond over here is the CD4 because T helper 0 cells are CD4 positive T cells. Okay? And then what's going to happen is, just like we talked about in viral infections, you're going to have B7 from the macrophage side and CD28 from the T helper cell side. What's going to happen is the macrophage is going to release IL-4. Okay? And then this is going to differentiate into T helper 2. This is going to be a Th2 cell. The Th2 cell, what it's going to do is it's going to release its own IL-4, so you have self-stimulatory signals, and that's going to result in clonal expansion. Okay? So this is starting to look familiar to viral infections. And now what's going to happen is your naive B cell, your Th2 cell is going to activate the naive B cell. Okay? The Th2 cell is going to activate the naive B cell. So remember, your naive B cell has an MHC2 on it. Okay. Your naive B cell, and by the way, it's not naive anymore actually. It's been activated by this macrophage. Okay? So now it has this antigen on it. That's going to be recognized by the T cell receptor. So the T cell receptor comes over here. This is stabilized obviously by the CD4. And remember, when you activate a T cell, it starts expressing CD40 ligand, okay? Which is going to find the CD4 over here. And then what's going to happen is you're going to release IL-4 to the naive B cell. So then what's going to happen is your naive B cell can go and it can become either plasma cells or it can become memory cells. Actually, let me draw the memory cells on the top because I don't need to talk much more about them. Memory cells and plasma cells. Okay, so we know that memory cells, what do they do? Memory cells are, they're just going to go, we talked about this before, they're just going to go into the bloodstream and they're going to exchange between your bloodstream and your lymph, and they're just going to persist. They're going to be specific for this exact antigen which is associated with this bacterial infection, and they're just going to continue to exist for the rest of your life. Okay? Then your plasma cells, what are they going to do? They're going to release antibodies. And again, these antibodies are specific for this disease. Okay? So now your antibodies have 3 different functions that they can do. They have 3 different functions they can do. The first function is you have a bacteria, and what they're going to do is they're just literally going to cover it up. This is called neutralization. Okay? Same concept as it was in viral infection. Neutralization. Okay? The second thing that it's going to do is it's going to opsonize it. Okay? And that kind of looks the same. Opsonization. Okay? But what opsonins do is they're going to recruit macrophages. So this is opsonization. And the last thing it's going to do is it's going to initiate the classical complement pathway. Okay? And this is the last thing we have to talk about, is the classical complement pathway. So now you have classical complement. And it's very, very easy because it's almost the same exact thing as the lectin pathway. Okay? The only difference is the first step. So essentially what you have is you're going to have, on your bacteria, you have antigen. Obviously because it's foreign, it's going to have its own antigens. And you just created antibodies that were released from the plasma cells specific to these antigens. Okay? The Fc portion, remember this is the Fab, is a portion that binds to the antigen. The Fc portion is the constant portion. Okay? The Fc is going to be bound by a molecule called C1. And that has 3 different subunits, Q, R, and S. Okay? This is going to do the exact same thing that lectin did. It's going to take C4 and C2. And because we talked about it about 10 minutes ago, I'm just going to quickly just write it down. Okay? So step 1, what's it going to do? You're going to have C4b. And remember, this is the exception, C2a, not C2b. Step 2, this becomes C3 convertase. C3 convertase, okay? So what it's going to do is now it's going to bring C3, and you have C4b, C2a, C3b. And this is called C5 convertase. And then what it's going to do over here, it's going to bring in C5, and then essentially what you have is C5b here, and then very quickly you're going to bring in C6, C7, C8, and then your MAC complex, which is C9. This is your MAC complex. And that's going to cause lysis, okay? And that's essentially all there is to your adaptive response. So just as a quick review, your adaptive response is initiated over here. Remember the macrophage that was the initial response in your innate immunity is going to come to your lymph tissue, and there it's going to do two things. First, it's going to activate your naive B cell. So now you have a naive B cell that's been activated. Okay, so you have an activated B cell. Okay, now this, in the absence of T cells, is going to be called T cell independent activation, which is very limited in what it can do. But in the presence of T cells, what's going to happen is the macrophage, in addition to activating your B cells, is also going to activate down to your T helper 0 cells. And the way it's going to do that is the same way it did in the viral in your viruses. Okay, is that your T cell receptor is going to find the MHC2, your B7 is going to find the CD28, and then the macrophage is going to release IL4. It's going to release IL4, which is going to differentiate your TA0 into TH2. And now your TH2 is going to interact with the activated B cell, and the CD40 ligand is going to bind to the CD40, not CD4, the CD40 on the activated B cell, and that's going to result in T helper 2 cell releasing IL4 to the activated B cell. Okay, so that's going to happen. And then the activated B cell can now become memory cells, it can become plasma cells, and by the way, it can also do isotype switching. Plus isotype switching. So you can create more than just one type of antibody now. And your plasma cells can go and do three things. It can either neutralize, it can opsonize, or it can activate classical complement. And we just talked about classical complement, which is essentially the same exact steps as the lectin pathway, except instead of starting with mannose binding lectin, you're starting with an antibody binding to an antigen and recruiting C1. And that's essentially all there is to bacterial infections. From the beginning, your innate response, where you have resident macrophages along with the alternate pathway, and then you bring in neutrophils and lectin complement pathway, and then you kick in your adaptive response. And that's really all there is. And there is just one thing that I forgot, and that is when we were talking about the complement pathways. You have all of these inhibitors. So you inhibit C3b, as we said earlier, you inhibit C3b with factor I, you promote the step to C3 convertase with factor D, you promote the step to C5 convertase with perperdin, you inhibit C3 convertase with factor H and DAF, and there's one more inhibitor you have to know, and that is that if you want to inhibit the MAC complex, what you do is you inhibit C5, C6, C7, C8 with CD59, okay? So if a molecule has if a cell has CD59 on it, it's going to prevent the MAC complex from forming. Okay? And that's essentially all there is to bacterial infection.